That's just because yesterday, I made an effort, to try to connect the disciplines, animal behavior, collective motion, synchronization. In the end, maybe like me, today you have seen things that go on, at the level also of different problems that can be more formal or more applied, in which you see the connections, right? So I was very excited today to see, about the synchronization that Jesus was discussing. And I see that Albert is not here because I wanted to start asking them one question. Imagine, they were showing to us, these nodes in the network, which probably you don't even know how it's made, how it's connected. And you see that there are these nodes that become to be synchronized. And at some point, Jesus was showing to us this picture in which they were seeing the number of nodes as a function of time that become synchronized. And that from there, it seems that this paper was able to infer a structure, not topology, let's say, the connectivity that you a priori maybe don't even know. So I wanted to ask Albert, whether they have to keep track of the number of nodes that are jumping and as a function of time, and what is the time that separates these transitions that we were seeing that were kind of jumps, little jumps as a function of time that are getting more and more nodes into the synchronization problem. This, for me, and it is related to what it is going to come now, is what we call cascades or avalanches. Okay, so this is the topics of today's talk. So that's why I was also nasty and asking Jesus that what if the time is also power law distributed, and even if you define an average, the average is not very meaningful. Because you can define an average, but maybe the average is just not representing the distribution of times in which things are happening there. Because you have long tails and things like that. This is the story that I'm going to tell you today, okay? So forget about this slide. This is yesterday's slide, but since the movie wants to start, I wanted to start showing you this movie that I just plugged in yesterday. And it's about these bees, so we reconnect again with the starting point. This is again some aphids, honeybee, and they are showing here this synchronization, and the reason that they produce and propagate these waves here probably is waves that are maybe more periodic, not the avalanches that I will be talking about today. But the eye is tricky. You have actually to pay attention, make a quantitative analysis, and see how many of those are actually participating and the time that it takes for this movement to happen. So here, what they are doing is actually a decision-making in this case of changing conformation. It's plain there, but they change their body conformation, and they communicate that to nearby bees in this swarm, which is similar to the one that I was describing the very first day. And that's because there is a wasp, one enemy, that is actually nearby, and they pretend to be more dangerous by performing this type of periodic motion and kind of a scary type of thing. So that's the mechanism that they have actually developed in order to scare the enemies that may come to attack, in this case, the swarm. Beautiful, right? We know that it is there. It has to do, again, with decision-making, the fact that in the end, they achieve this consensus to do that. They, in this case, it's not something that it is just stationary and it won't be effective. So they keep on having and propagating these waves. And again, this is a nice way to connect with this idea of criticality, because the idea is that if you are sitting close to a critical point, you will have this kind of behavior. And this kind of behavior maybe facilitates you this transfer of information that it is needed in order for the group actually to synchronize this way. Albert, so that was for you. I prepared it for you. So this is the bees, but now, exhibiting these waves, synchronized. It's like the fireflies, but in a different context. Here they do that just because there is a kind of an enemy, a wasp, that it is trying actually, to kill or to eat or to, get their resources, sorry. Nice, no? So I was trying to motivate that you have these behavioral waves in this case that now we are going to continue seeing in different types of groups. So today I was more talkative, I was not so prepared. Anyway, so let's go. So another example, before we come to the fish. And this is something that already was published in 2015, if I am not mistaken. I usually have shown this movie several times. You can download it from PNAS. Here there is this grazing sheep, in this enclosed environment, no predators, no danger around them, but still, I mean, these movements are very heterogeneous and intermittent. Yeah, this is speed up. I mean, this is a video that has been speed up. But the idea is that they slowly diffuse while they are eating, but at some point they just run and jump and cluster together. And this is the type of signal that you will see in the area, this like the one that we were discussing yesterday, remember the hull, the convex hull area. So you do that, so it's the area that contains all of them, and you see how this area is changing as a function of time, and this is the type of signal that you will see. Here is not predators. It's probably just that one, I mean, they are social gregarious animals. They like to stay together. You are eating a bit as absent-minded. I will do that, absent-minded eating. And then suddenly you feel that you are lonely. You are no with your conspecifics, and you start running. And this running actually is triggering a run in not necessarily all the group, but in some part of the group in such a way that they have these rearrangements in the area that they occupy. And also you could see similar things if, for instance, you look at the average velocity and things like that. Okay, so that's a little bit the topics of today. Okay, so it's the search for food, perception of threat. Just because you are scared, you have this behavioral shift, because you need to, like move a little bit more, can trigger what we call these behavioral avalanches or cascades. It depends on the context. And what I'm going to show you today, that most of the times when the systems are prepared close to this critical type of condition, these avalanches are not going to have characteristic scales. They are going to be what we call power law distributed. So that's why I was asking a moment ago to Albert whether, the nodes that become synchronized and the times at which these more nodes become synchronized, whether, they are distributed according to what? And he told me it's depending very much on the structure. But if you can use the activity, the synchronization, in order to infer the structure, it will be very nice also to try to connect with these distributions of sizes of these cascades that maybe are being produced in there, also with the times that you have in there at which you produce these jumps and things like that. And maybe, it's speculative, but I had this idea while I was listening to Jesus' talk today. So very nice. Okay, so now we continue. So the questions again is whether this phenomenology is just unique of some animals, if it's no more universal, what are the mechanisms, again, that are allowing that to happen, and whether you can model that in simple, maybe models such as the ones that we've been seeing in the last days, and so on and so forth. So there is also maybe the necessity to incorporate, again, the leadership thing, that you should now know that it is not a permanent thing, but can be just transient. And in this case, maybe just this sheep that was eating felt lonely and started running, calls the attention because it's changing its movement patterns. It's just calling the attention of everybody else's. And they can see, probably in this fence, there are long distances, but it is not that long for them really to see that something is going on. And maybe this startling that starts the thing is important for the triggering of the activity, that it is going to continue in time for a while. Okay, so these ideas in your head, but we don't have sheep. Well, we could go maybe to the field, but it's not so easy. We have the fish. So we decided to see whether, our fish, the fish tank that you already know, regarding, for instance, these strong changes in the movement patterns, such as in particular, now we are going to discuss the change of orientation, such as the omega that, Jesus will be describing in his talks for the oscillators, are triggering these behavioral cascades, okay? Whether one change in the orientation pattern of one fish actually is being followed by everybody, just a few, whether, I mean, the number of followers after that move is something that is well-defined, is if it's everybody, if it's just a fraction of it. So in a way also, it's a kind of a response to Amelia's question the other day. So in here we are seeing a little bit how these changes in the movement cues are being a little bit propagated in time. It's not maybe the same that you were asking, but it is some sort of inference of the time that these things are being are happening in there. Today? Oh, no, you are going to see here, power law distributions. It's not that you have, some part under heavy tails, but you will have just a power law everywhere, yes. And the exponents are heterogeneous, in the sense that you have values close to 3 that today, Jesus was discussing, but below, a bit below. So if you want to call that, a Levy flight, I wouldn't say that. I would say it's just power law, full power law, not just the tail, everything. Actually, the tail has problems, because of the statistics. It's full power law, okay? So we are going to see that in a moment, and the first thing that we did was to pay attention. This is even something that we did before we did the job, the work that I explained to you yesterday about the model for the alignment, selective, and now this we did before, a long time ago already. And the idea is that we noticed that one fish and how many fish you had in the swimming pool, we were characterizing the rate with which they were turning, this turning frequency, and it was pretty universal, so it doesn't matter. Well, it's true that here we are averaging over all the time and all the experiments that we have performed with different groups, but no fitting in here. So everything falls on top of each other. So it means that we have a very nice distribution of these frequencies that today Jesus also was talking about, that it is like that. So the first part of this plot actually is describing that they most of the times will just go straight. Remember the glide phase in which they just let them go. Once the fluid is dragging you and you become almost stop, then they will make a sudden move and they will rotate and repeat the process again and again. And this is happening whether they are alone or they are in the group of different constituents. And this is what we get. So it was nice to see that we had frequencies that were distributed like that. The flat means going straight for some time, and then not so much going straight, but I mean this decay, that it is something like maybe a power law, but with an exponent that it is in between. If maybe I'm gonna say something that it is wrong because I don't remember. It is not true too, for sure, because we were having some discussion about the Lorentzian character of this, but I prefer not to say it because maybe I'm going to say a lie. Maybe it's faster than 2, okay? But I don't remember by heart. Okay, so this is what we see at the fish. So every group will have this tendency to rotate this G of omega that Jesus is describing according to this form. And here is terms that are happening that are not bigger than usual, than the going straight thing. And actually, we put a threshold here around maybe 0.1, whatever, and you will see that this is what we call triggering events, in this type of maybe reorganization that we are going to see at the level of the group. So far so good. Okay, so now we continue. Well, here it's just, again, the same. So the idea is that these fish are rotating. We have this frequency that we define this way, but you could simply look at the change in the angle per unit of time, and that will depend on the resolution of your movie, but it is the same as this that I have here, and we are going to threshold our signal. What does it mean? That maybe there are some, they don't go straight all the time. It's not completely straight. They have very mild variations because they are moving in the water, but then below that, there are turns that are not that small. Some of them will happen because, of course, you are facing the wall and you have to turn, otherwise you just smash into the wall, That's something natural. Okay, so this is our signal, and now we are going to define a cascade in the following way. So by observing these rotations, the idea is that if they want to stay together, they will have to rotate together. But some of them try to rotate and nobody will be following them. So some are more effective in making this rotation something that like a contagion process, okay? Whereas others are more successful, and you will have these cascades that actually will be distributed according to these power laws that I was just discussing. So how do we define a cascade? Because we need to be together and understand what is a cascade. A cascade for us is the moment in which my signal for every fish, I track every fish, I have all the data, is rotating with an angle, with a frequency that it is higher than this threshold, I will consider that it is an active fish, active, okay? And then I will count at later times how many active fish do I have. So maybe this fish is rotating and, making that the other fish around him, because they imitate, they try to stay together, are going to do exactly the same. So I will say that an avalanche has a size and has a duration. And there are also inter-event times. This is the language that you will also frequently find in the context of earthquakes or other type of avalanching systems that you see in nature. So the idea is that you count how many fish are active after one actually started being active. You count how many of them, and that will be adding up just to give you the avalanche size. When nobody is active, because that's it, everybody is moving again, like flat, kind of smoothly, like a straight line type of motion, then we will say that the avalanche has finished. So from the moment it started until the moment it is finished, I will say that this is a duration, the duration of this event, this triggering event or avalanche or cascade. And then I will also take a look at what is the inter-event time that separates avalanches of different sizes. It doesn't really matter. And we are going to look from the data, all of these quantities, and we are going to show you how these quantities are statistically distributed, because it's going to be something random, but the distribution tells you a lot about what's going on, okay? So this is what I'm going to do. So yeah, this is what I'm explaining in this slide. Okay, first of all, before showing you the distributions, let me show you that we can also say where exactly we had what we call the initiators of this cascade or avalanche. Like in earthquakes, you can detect whether you have the epicenter of the avalanche here, there. In the space here is in the group, in the group of fish. And here I'm just representing the group of fish with respect to the center of the group, which, remember, is the centroid. So the blue points in this picture are the total accumulation of all these avalanches that I have. And although different colors represent different probabilities of that fish that it's in this conformation triggering an avalanche, you see that the higher, the points with a higher frequency to initiate these avalanches are where? In the periphery of the group, of course. Those fish that are in the periphery of the group have higher chances because they have more room, more space to really make a turn that it is larger. And actually probably are the ones that are more aware of what's going on outside. So yeah, that's not very surprising, but we actually corroborate it here. But maybe what it is more surprising, because if I had asked you before showing you this result, maybe you could have told me that, well, if you are close to a wall, the ones that see the wall have higher tendency to initiate the avalanche, so that we are going to see some sort of asymmetry in the distribution of these initiators in the position of the group. And we don't see that much. Okay, so an avalanche can be actually triggered in the, here, by the way, I didn't say, it is centered in the center of mass, the reference frame, but also is oriented, I didn't say it, with the motion at all times, so we reorient the plot, let's say, along the direction of motion of the center of mass. So the y-axis will be the velocity at each instant of time. So if I had that, I should see that the initiators are maybe more concentrated on the top of the y-axis. Why? Because they are in the front of the movement, in the front of the group. whereas we don't see that much. I mean, it's scattered everywhere. And actually what we see is that these avalanches can be triggered both in the front of the group, from meaning with respect to the direction of motion, then also in the back. So some fish sometimes that can manipulate, let's say, the group headings just from the back. Okay, it's something like that. This is what we see. Moreover, as I was telling you, in order to trigger avalanches, here I am representing the velocity that the initiators have at the time that they trigger the avalanche. Whether they do that when they are running very fast or whether they do that when they are running more slowly, right? What do you think? What you see here is the turnings, this triggering of these rotations are happening when the speeds, well, still distributed, but below the average that it is what I am signaling here with this red line. This is the average velocity for the whole duration of the experiment. And the triggering events happen when the individuals that trigger are going more slowly. And this is normal. As I was telling you, we glide, that's it. I have the decision-making process of where to go next. And at that time is where I rotate and I trigger these avalanches in the behavior of the whole group. Okay. You can also correlate, this is the speed, as we were discussing in the previous, the speed of the center of mass as a function of time. And big avalanches here are classified according to their size. Bigger are the darker ones. Usually happen when the velocity of the group is also lower. And little ones can also happen. Little ones meaning the ones that actually don't involve too many individuals. They can happen also in other conditions of mobility. This is what we see here. But let's come to the, sorry. So, yeah, when we were submitting these papers to the journals, so it's normal. We were always getting the question, no, but everything is due to the wall. But even if it's due to the wall, come on, doesn't mean that it is not interesting. Because you can rotate because you are reaching the wall, but the way you rotate is non-trivial. So tell me why, this kind of rotation is there, right? And I'm here just telling you that it is true that close to the corners in particular of the container, which is what you see here, here we are representing the position of the center of mass of the group when one avalanche has been triggered, okay? Where was the group within the container, okay? And big avalanches, again, which corresponds to these darker colors, are located more on the corners, of course, because from there they have to escape, so they will have to turn more. But other avalanches are more broadly distributed everywhere. So you don't have to be, so negative. And on the other hand, actually the activity, so the triggering density of the triggering density of events, let's say, is also very much actually is a bit higher when you go inside than in the corner. So in the corners you have to do it because you have to do it, but sometimes these avalanches are also being produced in the middle of the tank where there is no need to rotate because the wall is coming, okay? So several things that we investigated just to answer the referees, which finally, got satisfied with the answers. And still, even if it's due to the wall, it's interesting because what I'm going to show you now is non-trivial. Okay, so this is no big avalanches. Big avalanches, of course, are not happening closer to the wall because, everybody has to rotate, and these are some examples. I'm showing you several examples. Okay, let's continue. Now, yes?
How do you separate those events? If there is a small group of fish that's turning into a snipe, and then other small group of fish that's turning into a snipe, they're closely related in time. How do you separate? Is it like manually, or
no, the definition of what we call a cascade is very simple. I don't mind if the fish are close or they are far. Well, usually they go in groups. They don't really split. But whenever I have a fish that it is rotating in the whole group of fish, that is rotating beyond my threshold, I will say that I am in an avalanche state. Well, maybe you can argue that maybe it's not just one. Maybe there can be two together coexisting. And we cannot separate that. So whenever we have active fish, independently of whether they are located within the group, I'm going to say that I am in the same avalanche. And I will finish this avalanche when no fish at all is rotating with a frequency that it is above the threshold. So quiet period. Nobody active. Nobody rotating beyond that threshold that we have imposed. And then you will ask me, what is the importance of the threshold? And if we are serious, we have to discuss that, and we are going to discuss that. Okay. Exactly. To the border. No, whatever individual that started the avalanche, no matter whether the position of that individual is, will trigger the avalanche. So as you are thinking, you had the idea that maybe the ones that initiate are more often located near the walls. And what I showed you in the picture that I was discussing before is not true. No, not true. I mean, they are widespread, all around the radius of gyration, let's say. It can be that they see it and they have this information that the wall is coming. It can be that, I mean, I don't know if it's a good idea to go back to show you the one fish or the two fish. Remember that I was showing you two fish movies? They do that. They rotate. And it's not because they are close to the walls. They rotate because they want to rotate. I don't know. They are playing. That's my idea. No? The idea is that it's like us. You are an active system. Actually, they swim. They get oxygen from the water. I mean, why should you go straight? I mean, I will have the same question to you. Why should they go always straight? No, they are there. I mean, the walls are closed sometimes, but not always. And they rotate also in the center. And actually, I'm going to show you that you can also do this analysis that I am going to do with only the fish that actually are kind of far away from the walls. And you will see that the results that I'm going to show you don't change so much. Okay? Actually improve the results because of what I'm going to comment in a moment. Okay? Good. So this type of analysis in the dynamics, if you are studying collective motion, is something that you can do in all sorts of animals. I will be very much interested in seeing if there are more examples in which you see that. And actually, you don't need to have too many. Okay, you don't have to. No, as I was saying to you, here we are going to analyze groups starting by 8, 16, 10, something like that. So actually, the paramecia, I mean, the way they rotate, maybe, I don't know, okay? Okay, first I wanted to explain to you, and maybe you can do with me, what we call a null model, because in order to say that your results are significant, you have to compare with a null model in which you know that things are not going to be, correlated in time and in size. So that's the way you do it. Maybe if you want to make a picture. But the idea is that imagine that you have processes of turning that are completely uncorrelated in time. That won't be an avalanche. No? If there are no correlations, no avalanches. How will these events actually that are just randomly happening as if they were Poisson processes will be distributed like? So for instance, the calculation that I can follow with you here is very, very easy. Here I'm calculating what is the probability that a fish is active in a given frame in the movie. So what I am doing is, okay, it's going to be something that it is, the distribution of, how do I calculate that, the probability of a fish being active? The idea is that it has to be rotating with an angle beyond the threshold, right? So whatever angle I start from the threshold, whatever the other angle that it has, will tell me that this fish, according to the probability distribution of the turning, the frequencies that I have introduced in the previous slides, you can calculate that. Do you agree with me? That's the probability of a fish rotating beyond the angle that actually tells me that it is active. This is the probability of one fish being active. Okay? Now, what is the probability that at least one fish is active in a frame? So it's the way, these counts, these numbers, these analytics is always done, is always the same. So the idea is that you calculate what is the probability that if it's at least no fish no, at the time t is active. What is the probability? Q is the probability of one fish being active. We calculate it like that. What is the probability that at time t I have no fish active? Y minus Q. None of the fish active at time t to the n. And now I want the complementary that at least one, can be two, it can be three, doesn't really matter. So it's one. What is that? You see? So this is the probability that at least one fish is rotating beyond the angle t, omega c at time t. This is what we call the activity rate, because this is something that we are doing at the level of each time step. This is the activity rate. Now, with the activity rate, you can do several calculations that you see here. For instance, we can calculate the duration distribution. Maybe if you want to have maybe some time to think about how to do it, how will you do that? Well, that I have no active fish up to time t. So Q, which is what I was calling here, this activity rate at the level of the group. Q is the probability that at least I have one active fish at time t. I have to do that t, capital T times. Same next time, same next time, same next time. And this is the duration distribution, because I have to have at least during a time t fish that are active according to the activity rate. This is what we are doing here. Q to the t. And the rest, and when that is finished, the next move is that I don't have any activity in the system. That's why you multiply by 1 minus Q. So this event is just Q, Q, Q over a period of t, and then 1 minus Q finished. The avalanche is dead. And then you just have to normalize, because it has to be a probability distribution. T is supposed to be a discrete variable. You sum over in between 0 and t, calculate the normalization condition, and this is already a probability that it is normalized. You can do that. Okay, this is the distribution that you will expect if fish were just turning in an uncorrelated manner. You can always write that as an exponential distribution. So the probability of the duration of uncorrelated rotations will be something that goes like an exponential. You know how to pass from there to here? Yes, no? So the idea is that here you're going to have something minus t, and things, with a characteristic time. This characteristic time is going to be, excuse me if I'm not doing it correctly, like that, but it is 1 over Q. It's just rewriting this power in terms of an exponential with a logarithm and making things to look like the way you want, okay? So this is what I mean is the distribution that you will expect if you had uncorrelated events, okay? Exponentials. So now that you are prepared, the same thing you can do for the inter-event times with the same type of arguments, and a little bit more sophisticated, but if you are interested, is in the appendix in some of our references. It's very simple also to calculate the same type of probability for the sizes of these events that I'm going to show you now, when they are supposed to be uncorrelated. So there are simple, probabilistic arguments that everybody can use and do. Okay, so believe me, and the relation in between what we call the average duration and the average time, and the duration, the average size of a given duration is something linear in this null model. It should be linear in this null model. These are all things that you can calculate. So now I'm going to show you what we get in our experimental data. So yeah, you have to take into account that the systems are not very big, so you are going to have finite size effects, because I cannot have sizes beyond the whole group, right? I mean, I cannot have an avalanche that incorporates more fish than what I have in my box. So they have to have finite size effects for sure, but what you see, according to what we are able to observe, is that the durations are power law distributed, at least for some part of the distribution, which is this that you see in a log-log plot. Not only the durations, but also the sizes, just counting how many fish are rotating after one has rotated. This is the size. And you will see also these power laws with these bumps, of course, for the tails. That's why I was telling you that the tails here are not so well behaved, but I will explain you why. And this actually is what you will see by changing the threshold, which is crucial here, right? So you will put a threshold, of course, not in the straight, part of the distribution but you have to distinguish going straight from turning, and that is going into the tail of that distribution. But you can choose different points where you put your threshold. And the idea is that, if you are doing something that it is robust, it cannot depend very much on where you put your threshold. And actually, in this context of avalanche dynamics, people have studied very well what is the influence of this threshold. Of course, there are studies that are a little bit more serious. Because these avalanches appear, if you didn't know about them, they appear in many different contexts, also in condensed matter, let's say, when you have disordered systems, the flow of a fluid through a porous media, they appear in different contexts, a membrane that it is moving over a rough substrate. There are several situations where the growth of magnetic domains, when you ramp up an external field, ramp an external field up and down, that it is moving and making these domains grow. In all these fields, you will find these avalanches with the same phenomenology. Okay, that's one thing. And then you do consistency checks, because I was telling you, so first of all, power law, non-exponential. Second of all, consistency checks. Here, once you have obtained these distributions, if you assume that you are just not worried about what happens with this scale-free region that you see in the middle, you can do some scaling arguments, but very simple, in order to relate again this average size given a duration with the duration, and you will see that you have to have this exponent m that is related to the distribution that you find for the sizes and for the durations according to this formula. This is theory. And now you see what is the value of those exponents. You can also just measure these curves in which you compare the average size for given duration. So you classify your avalanches according to the duration that they have, and just calculate what is the average size that those that have a given duration have. And you represent this, and it's a consistency check to see that these exponents that we were measuring make sense, because this is another way of making them, of corroborating them. And you see that this is where it was expected to find. Well, it's okay. And this is for one threshold value, and for another threshold value, you can increase and decrease a little bit this threshold value in between these values that I am representing here, and you will essentially find the same. But it is true, if you ask me, if you increase too much the threshold, you are get out without nothing, because you are not going to have events that are implying turnings that are much bigger than what you see in your experimental system. So you cannot really go to very large values, because you won't have activity, you won't have avalanches. On the other hand, if you decrease it too much, then you won't be able to separate one avalanche from the other, because the activity will be continuous in time, which is maybe something that also was worrying you. So you need to slice, let's say, your signal into little spikes. And this is also normal, because here we are not dealing with a discrete system. The fish will continue moving with little variations in their rotations. But it's never the case that they are gonna, like maybe in a model avalanche, it's never gonna get freeze, completely freeze. Never, ever, never. Okay, there was a question here.
Normally, the scale-free phenomena is at last two scales. So, because
2 scales, you mean? No scales.
2 scales, like 2 packets.
Yeah, here is not very large. I agree with you.
So it's not a problem when you...
No, it's not a problem if you don't have better than that. It's not a problem.
You're looking just at the specific reason that
The power law, yes. Yes. Here is 1.5, maybe close to 2. But it's clear that it is non-exponential, because we can compare, and I don't know if I did, but maybe it's coming, with the null model. You will see how the exponential distribution will look like, and it is very far from what I am representing in here. Okay. Maybe you are not very convinced, but you have to propose me another model that explains better what I see. So that's the idea. Okay, so here it is. Also for the inter-event, like in earthquakes, they will be also distributed according to these power laws that are not incredible, but they are there, and they are not exponentials. And this is, again, with the same 2 thresholds that I was explaining to you a moment ago. By the way, you remember the null model that we were doing together, from which we were expecting these exponential distributions in here? What matters is the activity rate. You can also calculate the activity rate in your experiments. What is the activity rate? Is the number of avalanches that you will have per unit of time. Okay, this is the Q that you will see experimentally. If instead of choosing the threshold by hand, instead you just plot the avalanches and the distribution of sizes and durations that you have, when you select the threshold in order to preserve a given rate of avalanche production as a function of time, then you don't have to do anything, and you will see, I didn't do anything in here, that they will immediately, this is the word that we use, collapse. So you see that preserving the activity rate with the threshold, actually you are changing the activity rate, right? Why? Because since the signal is the way it is, you put the threshold here, and maybe you are just left out with some spikes that go above the line, above the threshold. So you are changing how often you have avalanches, which is what we call the activity rate. If you change a little bit the threshold in order to guarantee that you have an activity rate that it is comparable, as a function of time and for the different experiments and repetitions that we do, then the figures actually will not actually even change with the threshold or this activity rate, but they will immediately collapse in this universal curve that you see over there. Okay, that this is also nice. I mean, something that you, wouldn't expect to have so nicely and easily here and cheap. Okay. And finally, here also you have the same behavior for the distributions of these inter-event times. Okay, good. Different exponents. I didn't pay attention to the numbers because still, as you said, it's only one decade and a half. So maybe who cares? These exponents, we are not there to make sense of them and to try to compare these avalanches with avalanches that happen in systems that have been studied in the literature for very long, and for which there are exponents that everybody will already agree that they are well measured. In here, we are far away from there. So if you do an experiment, if you have a tank in your university and you do the same experiment with many more fish, maybe this exponent is going to change a little bit. And I'm not angry because of that. It's normal. If you have a finite system size, your exponent is going to be not very well measured, inferred from those curves. But this is one thing. Another thing is saying that, it is not interesting. I think it's interesting. Okay, so not only that, maybe this is a little bit more technical, but there is something that it is called the Cessna scaling. Yes, please. It's okay. Well, this is a hypothesis that we will be using, and I already mentioned it in the very beginning of the talk. So remember the decision-making process. There is some signature of criticality when we were talking about the bees. In the synchronization, I don't know if they have avalanches. Maybe they do. Maybe it depends on the kind of connectivity that you have in these networks that today we were seeing. But the idea is that if you are in the neighborhood of a critical point and maybe your system is able to, by changing this type of activity that they have for the rotation and so on, maybe they are operating close to this critical point, and this is the best explanation that we have so far. So close to critical points in out of equilibrium conditions, the appearance of these avalanches is something that it is normal. But it is just a hypothesis. Nobody has demonstrated that this is the case. Well, in some cases, in physical models, you can do that with the models. See that you have the critical point there and see what is the phenomenology around, and you will see the avalanches. And there are even universality classes, and the avalanches will have to have given exponents. We are not there yet, but we see them, and maybe the hypothesis behind is that they are close to a critical point. And why? Because being that way, excitable that way, with these correlations that are not well defined at the level of promoting these cascades of behavior, is the way in which you transpass this information more efficiently throughout the system size. Sorry, okay? And yeah, this is the Cessna scaling, and this is demanding, but we, in the end, we are able also to test that there is this scaling that Cessna proposed for avalanches in different contexts. The idea is that you have also universality in the shape of these avalanches. This is the curve above the threshold. It has a particular shape. If you get those avalanches that are scale-free and you order them and rescale their duration, the time by their duration, and the size also with these scaling properties that you see over here. And it's just the number of active particles that you will have as a function of time when our avalanche is being produced. You will see that you have this Sedna scaling, and even the fish are also obeying that, okay? So good. We have these signatures, and we were happy to have them. And as I told you, several people will tell us, oh, it's the walls, it's the walls. Yeah, it's the walls. But even if it's the walls, why the response is like this? So despite that, I mean, we also wanted to see what happened if we were only counting the avalanches that were happening inside the tank, so not so close to the wall. So this is what it is represented in here. So I'm just going to count the activity and the avalanches that happen when fish are located, separated from the walls. I think it's not so important, to tell you the truth, because they can see the walls even if they are here. But, just to make the people happy, we did that. And the power law is much better now. It's not that you gain too much, in orders of magnitude, in scales, in decades. But the bump that you were seeing before has disappeared, or is a little bit milder. Why? Because at the tail of the distribution, actually, we have avalanches that have a characteristic size. And these are not all the fish participating in the event, usually associated to the fact that you are very close to the wall, and everybody has to rotate. So the avalanches that are big and that are not concentrated on the corners of the container actually are not the ones that are in this scaling region. So, well, we have them, yes, but they are not the interesting ones. The interesting ones are actually the ones that happen at the intermediate time and size scales, okay? And this is just a little bit also of a demonstration. Well, my last exercise, if you won't continue to continue working this evening, is that I propose you already to code the Big Sec model, to propose this variation of the velocity for the fish, and so on and so forth. Now, with the very same model, if you did the effort, you could play a game, and it's just that you have a random, a particle at random that you choose at random in the system, that it is performing this kind of a time of a run and tumble dynamics, which is what the fish essentially are doing, but in a correlated manner, and that is just rotating above the threshold, let's say. So it's rotating more than others, that others will do. But at that time, when it does that, I am proposing that in this Big Sec model, you consider that it is making the role of an effective leader, a transient leader. So everybody, it's true that it has to be everybody, or almost everybody, at that time is going to integrate it, no matter whether they are, this particle that it is rotating above the threshold, that you choose at random, is within the interaction area in the Big Sec model. Remember it, we were discussing it as an exercise yesterday, right? So the idea is that if you do that, and you take into account your neighbors, you align with your neighbors, but you also align with that one that actually you choose at random, that it is making a random rotation, that it is above the threshold, you will be able, if you repeat this many times, you will be able to trigger avalanches in this model as well. But the exponents won't be exactly the same. You have to work hard in order to get the statistics, but you can do that. And this is a model, simple one, that you can use in order to trigger avalanches. If you don't do that, in the Big Sec model, you won't have power law distributions. No, you don't have that. So it's not easy. So I'm telling you, maybe the decades are not much, but it is a phenomenology that it is related to the correlations in the dynamics that the fish have. Because with simple, let's say, scenarios in which the correlations are maybe short range, such as in the Big Sec model and so on and so forth, you won't get this phenomenology. You won't get these distributions. So it's much easier to not get them than to get them. You have to think a little bit on what you have to put in there in order to get that kind of size and duration distributions for these events that we call avalanches in here. Okay, but maybe there are other models in the literature that can appear at later stages in which you have the same thing. Yeah, well, Not every time step. No, sorry, no. So the idea is that you choose one, it's rotating a lot, see as a consequence of the dynamics that it is natural in the model, whether you will be, by imitation of that one and whatever you have around you, you are continuing with the avalanche because you have active fish that are rotating above the threshold. And only when you finish one of the avalanches, maybe you can do it also without waiting, but the idea is that you wait. You don't overlap too many events altogether because then probably you are not going to see the same phenomenology. So you wait. You can wait maybe for given windows of time, but the idea is that you have to wait until one avalanche has passed in order to trigger the other. Okay? It doesn't have to be periodic. You can wait, or maybe you can just start a little bit before. But if you start all the time doing that, you won't see this phenomenology. So there is a separation, what we call a separation of time scales. See? More or less clear? Okay. So this is the exercise the last exercise that I wanted to propose to you. If you are interested, it's a good thing to have these models. And well, maybe you tell me, oh, Carmen, but now you are including a particle that everybody is seeing. So if it's very crowded, maybe it's not a very good hypothesis. But I'm showing you that, in the type of system that we are doing, the long-range vision that is in the system will make this hypothesis very, nice. And I don't think that it is going to have any problem, no? No, I don't have any problem in accepting such a hypothesis. What I'm showing to you here, I don't know why. What I'm showing to you here is another experiment that I promised you, in which I had now the tank, which is separated in between two compartments. In here, the compartments are separated in a way that, the fish cannot cross. And I'm just telling you here that there are very different behaviors depending on whether the wall that it is separating the compartment into two pieces is transparent, so they can see through, when it's opaque and they cannot see through. So just to demonstrate that, that is very important, the vision, and actually the correlations in between the behaviors in the two parts of the container when you have a transparent and opaque wall in between are very different. Here, for instance, I'm just representing what is the distribution of this velocity, of the center of mass. For the case in which you have a transparent, the distribution, sorry, this is the angular difference, I forgot. It's the angular difference in between the velocity of the center of mass of the two subgroups. So we have one group that it is moving. I am characterizing the motion of the center of mass of the centroid. I am doing the same thing in both sides of the tank. And I am actually checking what is the difference in between the orientation of the center of mass of the two subgroups. And you see that the difference is around 0 when the wall is transparent, whereas, it's very much, actually even counter-oriented when you have actually an opaque wall. So it means that, the vision is very important in these systems. And also here, I'm just characterizing, for instance, what is the distance to the wall, because they want to be together, but there is a wall, and they cannot so much as they like to. So they concentrate very much around the wall in one case, whereas, it's not the case when they don't see each other. So the vision is very important in this problem, and this vision actually will be, a way of arguing that having this effective leader that everybody can see, no matter whether it's within your Voronoi neighbors or whatever, is going to be a good idea. If it's exhibiting strong movement cues, that's the point. So he has to, or that individual that is going to act like an effective leader has to exhibit some strong movement cues, either accelerating velocity higher than the rest. And in this particular case, what we are actually doing is when it's rotating above a threshold, which actually is what we have used in order to define these cascades. Okay? Good? So this is just a justification of, and if you do it, but you have to work hard, you will get avalanches just with the big check model, but the exponents are not very similar. And here the system sizes are much bigger. So yeah, maybe the experiments will give us these experiments if we go to much bigger sizes, or maybe the model is not very good and you have to do something else. Still room for you to actually, if you want to investigate. And well, continuing with this topic, because I didn't want to change topics, not today. It's the last day, already 4 days. You maybe are tired to see me often here in their classroom, but I wanted also to explain to you, if you are interested, that there are also spatial and temporal correlations in these avalanches that we see in fish, and that we are going to use, and we are able to use actually the techniques that the people that are working on earthquakes are using in order to estimate these correlations that happen simultaneously in time and also in space. You were asking, right? I mean, there I was not caring very much about whether the fish were close by, the ones that are triggering, or how is the avalanche developing in space, let's say. It was just a matter of seeing how it develops in time. But now I'm going also to take into account whether I have what we call a main shock and an aftershock. This is something natural in earthquakes, right? So after a big one, you have heard the news about Santorini, maybe. Maybe you have small ones, and also what we call foreshocks and main shocks. So the idea is that if you have movements that are not very big, people know that there are no high chances that you are going to get a big one. So foreshocks, main shocks, aftershocks, it's just ways of referring to correlations in time and also in space. So they usually happen in the same area. So if Santorini had some foreshocks, maybe they are expecting to have main big one again in Santorini, not somewhere else, around Santorini. Okay, so this is something that you can also apply to avalanches in different systems, and also in particular to the avalanches that we see in fish. So the idea is that with this, here you see the plot. So there is also the background activity that is separated with my threshold. I don't care about events that are very little ones, because maybe it's just natural that you have always there some activity, but I'm going just to pay the attention to the ones that are beyond a given threshold. And then I'm going to classify it as a main shock, the big one, and I'm going to actually be able to see whether others that are coming in the sequence in time are going to be children, offspring of the ones that were higher and were occurring before in the sequence in time. Okay, this is what we did. And actually, there is one measure that the people in seismicity is using. It's called the proximity. It's just a way, as I was telling you, of incorporating both space and time, and also the intensity of the size of the avalanche that has occurred, in order to, in a sequence that it is ordered in time, see who is the predecessor of that other event that you see as a function of time. So you can classify now whether you have a main shock and several aftershocks, and see who is actually the predecessor of the aftershocks that you see as time progresses. Okay, this idea. And then I'm not going to introduce you all the details, because they are technical and maybe you are not so much interested. But one thing that you have to take into account, that it is different here than what you will have if it's in the open earth, is that the fish actually do these triggering rotations. They trigger these rotations and they spread these orientations, but that they are closed in the tank. So it's not like in the air. So the idea is that maybe you trigger something that it is in your neighborhood and is correlated with what was happening before. But if you wait too long, the fish actually will have to come back and pass through positions that they have already visited a long time ago. But that doesn't mean that, the activity that can happen at very long times after something happened is going to be correlated just because it happens close by in space. So we have this, again, boundary effect that unfortunately we cannot remove. And you have to take that into account in order to make sense of what you see. If you do that, and you are careful and estimate the times over which actually the fish go around and do several rounds, they go around in the tank, you can see that there are these correlations that are in space and time. And there are these bimodal distributions for these joint distributions of size, or the presence in time, the fact that you have them, the earthquakes in space, and also the time at which these aftershocks. So I say again, because I was not very good. So the idea is that you have the main shock, the aftershocks, you have classified them, and now you see what is the distribution to find those aftershocks that are happening close by you at different separations from the place where you had the main shock. and also at the time, no, the same correlations at the level of time. But you cannot go very far away in time, and of course you cannot go, by obvious reasons, very far away in space, no, here. But still, it's very clear, if you randomize the data, this is what you will get. No, these levels that you see here with different colors is the data, our data randomized in such a way that you destroy all the correlations. And this is not the bimodal distribution, that, by the way, is also something that you see in earthquake dynamics. So there are usually bistable distributions with these two, bimodal distributions. And the one that it is meaningful is that one here. And this is nice because everything is concentrated and exhibits these correlations. And not only that, you can also try to come out with this idea of whether you have what it is called in the language of earthquakes an Omori law. Omori law, everybody probably have heard about this Omori law, this probability of having another event after a given time once has happened. So for earthquakes, actually, there is a power law that tells you that there is this distribution of the times after which, after a main shock, and the probability that you are going to have an aftershock given some time. And this is the Omori law, but the Omori law there has an exponent that will be close to 1, whereas in here, just because of the size limitations that we see, or maybe because the fish do it in a different way, we have a stronger, bigger exponent of close to 2. Okay? But you still see that these temporal correlations there decay again like a nice power law. And well, I mean, if you don't want to believe it, it's okay, but we have now done experiments with different system sizes, many experiments, and it seems that it is very much consistent, and still we don't have maybe a very good model to explain that, but we hope to continue progressing along this line of research and maybe improving and connecting actually what we do with what I was telling you yesterday about the selective interactions and so on and so forth. So there is a lot of still work to do, and we keep on doing, okay? So that's for this part, and I just wanted to not change topics very much. The last part is that I'm going to show you these also experiments that I was telling you that we are doing in this compartmentalized tank. And it's not disconnected. I mean, in my head, usually things are very much connected. I don't like to do research that it is disconnected. And my collaborators criticize me because of that, because I have this necessity to connect things, to make the pieces of the puzzle really, coincide there in the table. So this is also, if you don't like the walls here, there are going to be more walls. But what I'm going to do is to try to see whether this activity that it is correlated, for instance, in the rotations that I was describing a moment ago now, is something that you can see when you have these two compartments in your tank. And the fish have the possibility of crossing or not crossing. Okay? So if that kind of imitation and the necessity and the decisions that they make in order to preserve the cohesion of the group is there, I expect to see also some signatures of that type of behavior in this experiment. Why? Because when actually one fish is passing, I don't expect that it is just passing and it's something that it is another fish is going to do at random, as if it was a Poisson process. I expect to have also the same signatures of correlations in time and of size in this experiment, where in here what I'm going to do is to count how many fish are passing from one side to the other. But for the same reasons, because they want to stay together, they decide to move in a way to stay together, and this is going to introduce the non-trivial correlations that are going to give me this type of dynamics. This is just a repetition, but you see fish crossing because we have a door. Yeah, you can change the size of the door and see whether they are going to be affected in their transition rates by the size of the door. And you can actually, I didn't have the time to analyze all the data that we have so far, because we are repeating more and more experiments in order to collect the statistics. You need to collect the statistics. And the tracking, already the techniques that we use. It's not easy, but we have many more data now, and I didn't have the time to prepare you updated versions of the plots that we are going to see in a moment. But you have to trust me a little bit, and I'm going to show you that, for instance, there are several quantities that you can measure. First of all, you see strong number fluctuations. We do experiments that are half an hour experiments. The duration is half an hour. And in half an hour, you will see different experiments, different ways in which the fish actually are trespassing no, the starting point, no, the starting part of the box and go to the next one. You can start with experiments in which everybody is in one side, half and half, and no, you can analyze, I mean, what is the dynamic like. You can count how many of them, no, are crossing, what are the times, no, along which, no, on which, no, the fish are crossing, all these types of observables. Maybe I'm open if you have some ideas or suggestions, no, to make me. I'm very happy to hear about them, because you can measure several things, several different things. For instance, in here, it's all there. Here the statistics was very poor, but I didn't have to redo that. So the idea here is that we are counting the time that a fish in each experiment, no, you have all the fish, all the trackings, so you can have an information, for instance, how long the fish actually spend, no, in the other side of the box. That, I mean, it's not the one in which it started. No, it will visit the other and will maybe come back at some point, and you can calculate the time it takes for it to actually go there, how long it stays there, and comes back. And this you can do it at the level of each fish, for instance. And you see these ugly distributions, but that look also very much power law distributed with an exponent that it is small, and then it is interesting. The power law is interesting. And this doesn't really matter whether, I mean, you do it with all the fish in one side, but you can also do it with half and half. That is not changing. It's a robust feature. So you see these strong fluctuations in their behavior. But if you put all the experiments that we have, no, so, well, that we had at that time together, you see that there is some sort of also kind of universality in their behavior. And these times that, that the fish spend in the other box, different from the one that they belonged to in the very beginning, you will see these power laws. But not only that, just to make the story a little bit short, here we are counting, the crossings, how many they cross in one direction. And we are interrupting what we call an avalanche when we have crossings coming from the other direction. So imagine, if you have every fish in one part of the container, they will start crossing towards the other part of the container. And we could count how many of them did that. But if you keep on counting and you don't stop time, actually everybody, more or less, I mean, it's not interesting. But the idea is that once they have crossed, they can come back. Or even if you start with the box separated in between two compartments with the same numbers of fish in both sides, you will have transitions from the left to the right and from the right to the left. So the idea is that we will call an avalanche. You have to introduce a definition. Maybe you don't like it, but this is the one that we actually found that it is giving nice results. And is that an avalanche will be finished the moment we go in the motorway, we pass, and I am stopping the avalanche when I have people coming from the opposite direction. And this is already, breaking the correlations in the passing of the fish from one side to the other. Does not make any sense that I put everything together, right? Because the movement that they might have from one chamber to the other doesn't have to be necessarily correlated with the movement in the opposite direction, because they are different fish, right? So I'm just counting an avalanche as the number of individuals that are crossing in the same direction until that event has been interrupted by others coming in the opposite direction. And this is what I am representing with these colors. So these are the events that are happening as a function of time. For instance, this is just a little piece for you to understand. But this is the type of data that you will have, and then you can count the inter-event times, the number of their same points, the number of points with the same color, which will give you information about what we call now the size of the avalanche, the duration, the same things. And so far, I'm here just representing to you what we get. This is for the size. Well, it's not bad, it's good, but the exponent is not very, it's big. For what usually are avalanches, it's big. Okay, this is where we are, and we are working in order to improve these statistics and see in the end if this is the right observable. This is not work in progress. Still, I don't have anything published in this. And other things also related to quorums, and we are investigating other stuff. And here I'm just comparing what we obtain with what you will obtain if you were randomizing again. You always have to control your data and compare your data with what we will call this null model type of result. And it's different from what you will get if you just randomize the events in time. So the correlations will be destroyed, and you will get distributions that are not the same, okay? So correlations are there, and this is the point, no? And again, no, power laws are there, and this is my point, that, I mean, there is something in between them, something in them, that actually allows you to see different dynamic features characterizing their movement and their decisions, which is, in general, what I told you that yesterday, that it is undergoing, no, in these effective forces, no, and effective models that we propose that have, no, this peculiarity, no, in these animal groups that want to move together, okay? So whether I was, no, able to really convince you that there is something going on, that it is interesting, that makes sense, that we keep on investigating or not, I hope, I mean, it's up to you, but for me, it's encouraging, and I like, no, this idea of, no, pursuing more research. This is just also something that we wanted to check just for the fun of it, and is whether, I mean, there are some individual in these groups that we have, no, up to 100 fish that have more tendency to cross more often, no, and from, no, that are the ones that are leading, let's say, no, that are the initiators, no, that is what we usually use as a word. So yeah, so this is, no, so random test that you have actually to implement in order to compare, no, for also, no, showing that your data are meaningful, and but by randomizing and just demanding that you have a 99% interval of confidence, you will see that still there are some fish that have recurrently a little bit more the tendency to cross. I don't know if this is something that it is going to be very meaningful, but it is also, no, again, a corroboration of this idea that they don't have to be same. Here is all the fish that we have in the groups, but we cannot see who is who in different experiments. Unfortunately, we don't mark them. No, we don't put them any clothes or any hat in order to see whether they are always the same in the same experiments. But what it is clear is that in the same experiment, in one experiment, there are some individuals that maybe because they have more information, they are more bold, whatever, they tend to do that more often than others. So the leader and follower idea that I was telling you about yesterday. Okay, so this is maybe not so close correlated with what I explained to you yesterday, but yeah, no, it's an indication. And I think that with that, it's 1/4 of an hour less, but I think that today I'm going to just finish. Tired for days, 1 hour and a half here with you. And first of all, well, these are my conclusions. So, you know, animals that move in groups will tend to have, you know, decision-making, no, mechanisms in order to preserve the cohesion of the group. As physicists, we just don't study behavior. We could just describe that with words. But the idea is that we propose simple or not so simple models. Remember that yesterday we were talking about people there using, no, other models that people will use in order to study neurons, in order to understand this synchronization, no, in their swimming patterns, in the fish swimming patterns. Then there are people that maybe could use, no, such as, no, Ignacio that is now in the audience, Albert, no, Kuramoto models in order to try also to investigate, no, this synchronization and relate that with their motion and their characteristics of their motion and distribution of this motion as a function of time. We can also propose these effective forces that are more, no, the traditional approaches that you will find in the literature in order, no, to have, you know, models that are able to describe the phenomenology and that are nice to us because we understand everything that we put in and what we get out. We saw that maybe a good way of modifying those models was to try to incorporate a little bit the idea that they are living matter and that they have these selective interactions, no, because they are more attracted to other animals, no, in these representations of these models that have, no, strong movement cues that make, no, those that you are going to follow interesting. Okay, so that was a mechanism that we exploited in order to get, no, some observables and reproduce some of the observables that we have in the experiments. I think that the idea is nice and is something that, I mean, in living matter is something that you have somehow to consider in the way you like it. These days, people will actually introduce what they call non-reciprocal interactions. It's another way of introducing that type of stuff, and it doesn't necessarily have to do with vision. It can also have to do with, no, vision related to strong movement cues, not just, no, to what you see. To what you see, but selective, even though what you see, but selective, okay? And then finally today, we saw that these dynamics, that it is triggered by the fact that you have these selective interactions of imitating maybe the ones that have these strong movement cues, such as rotating higher or going faster, actually is giving rise to this dynamic response in time that it is non-trivial, but in the form of these behavioral cascades. So, the pieces of the puzzle, at least in my head, are, now becoming not to go on top of the other. No, maybe for you still there are some gaps to fill in, but we are working in the direction of trying to fill the puzzle. And this is what I discussed last, and with this not only temporal correlations, that maybe there is also something that you can find in synchronizing systems. And I'm going to, for sure, think a little bit more about this idea of inferring structure from the dynamics that you see, although these networks are not going to be very complicated and power law distributed, but still maybe interesting. And this is just maybe for the sake of these groups of animals transfer information in an effective way to maximize their efficiency, which in this case should essentially prioritize the fact that they stay together, not the cohesion of the group. Because for them, it will be different for other animals, but for these animals, staying together seems to be the rule and more than the exception, okay? So this is all. So I also had this slide, like in the first part, remember, in which I was suggesting to read only a few papers, not during the school, not at home. At home you can read hundreds and thousands more, but a few of them that I have used in my presentation. And also our references, of course, I have to advertise also the work that we did. So they are. They have been appearing along the talk, and you have the recordings for you to actually go over the details. And let me just thank you very much for being here until the end, 4 days together. I had also very nice interactions with some of you. I hope that we continue these interactions in the future. I look very much forward to it. And with that, I finish also thanking, I had also introduced in the different parts who were my collaborators. This is just the whole group with new students, some collaborators that are frequent collaborators, let's say, very permanent, persistent collaborators, and some of the people that are maybe appearing into the plot and actually making us think with even more ambition into the problem, and hopefully many more, okay? So thank you very much for your attention, and I am open for your questions as usual, as every day.
Thank you, Carmen.
Welcome.
Some animals or even some fishes have handedness, even if they don't have hands, but they have a laterality. They have a preferring direction to
we don't
rotate.
see it here. We checked that. When
Oh, you don't see that?
When I was plotting the distribution of the frequency of the turnings, remember? This P of omega. Now I just represented positive, but actually positive and negative are completely symmetrically distributed.
Even if you put the wall between them?
Yes, because there are walls everywhere. So
even if they are trying to be closer with each other, but it's different parts of the wall, so there are.
Yeah, but
no difference?
take into account, maybe I could look in windows of time, depending on the relative position of the group to the wall, and I will see what you have in your head. But this is averaging over the whole duration of the experiment, and the fish are not, getting localized in one wall. So they move around. So when they are here, they will have more tendency to rotate to the right, but when they are here, they will compensate that with rotating to the left, and you don't see that, the breaking of symmetry. So they are natural rotors, but they will equally likely rotate towards the left or towards the right, at least these fish. Although maybe I have to, check very carefully individuals one by one, and, in order to answer your question,
okay, thank
with
you.
all the knowledge, I will have to do that, and I haven't done that. I have done it, statistically over the duration of the whole experiments. Okay.
Okay, thank you. I have a quick question. How general are these behaviors? Because
How say?
General or
General.
like global, universal, I don't know, because
Yeah.
you have been making all these experiments with a single species, right?
Yes, you're right. I know that there is a paper by people, by the way, I think in Brazil. Already I forgot the name. Sorry if some of you maybe are here in the audience. I was not able to have it in my head. But I know that there are other people that have repeated the experiments and they copy very much what we have done in our paper. So if you look at the papers that cite our works, you will see that there are other people that have already found these avalanches. Also in other fish.
Cool.
Repeating the same type of analysis that we were doing in our papers.
Cool. Thank you.
I forgot, but I can find it for you if you want. At least that, and maybe more coming, no? Who knows?
Is there any other question, comment?
Come on, go ahead.
Come on. Okay, if not, I think we can thank again
Yes.
Carmen for all these lectures and the hands-on. Thank you, Carmen.
You're welcome.
And now I suppose you are hungry.